All right, welcome back everyone for more human physiology. So, now that we have kind of covered the introduction to the somatic nervous system, we can start talking about each of the special senses. And we are going to start with what I would consider to be the least complex of the four special senses. So, we're going to start with taste and smell, called gustation and olfaction. So, let's get into this. So what we're going to find here is that taste and smell operate almost identically to each other. There's just going to be some anatomical differences between the mouth and the nose, as I'm sure you would expect. So taste is a sensory modality that is going to be mediated by activation of chemoreceptors. If you'll recall, chemoreceptors are sensory receptors that are activated by chemical ligands. So really, the activation of sensory receptors that we're going to see here is going to be an awful lot like those ligand-receptor interactions that we saw in the communication unit. So in the case of taste, the ligands that bind to receptors are food molecules that dissolve in the saliva of the mouth. And as it turns out, dissolving the food molecules in the saliva is absolutely required for any signal transduction to occur. We have to have the saliva in order to get the ligand in proximity to the receptors. So try taking a paper towel and drying your mouth out next time you want to eat something. When you do that, you're going to notice that your taste is pretty muted. So you got to have the saliva in your mouth to really be able to taste things. So there are 5 submodalities or individual tastes based on the ligand that activated different types of receptors that we're going to see here in a minute. I'm sure you're familiar with most of these. So we have sweet, sour, salty, bitter, and savory or umami tastes. So each of these, like we say, is activated by a particular type of food molecule. And most of these kind of make sense, right? So the sweet tastes are going to be activated by different types of carbohydrates. And different carbohydrates kind of have a different sweetness grade to them in terms of how well they activate those receptors. Sour tastes are going to be activated by acids, and if you go back to chapter 2, our definition of an acid is a substance that increases the hydrogen ion concentration in a solution. So when you eat something that is very acidic, all those hydrogen ions are going to activate a different type of sensory receptor and cause you to taste a sour taste. Salty tastes are activated by sodium ions, which makes sense, right? Bitter taste is actually interesting. This is activated by a select class of compounds called alkaloids. I'm showing the structural formula for a type of alkaloid compound that I'm sure all of you are familiar with and you have all probably ingested at some point today. So this is caffeine. So it's the reason why, for example, coffee tastes a little bit bitter. So the bitter taste is actually kind of has an evolutionary benefit to us. Basically, the idea is that early on in human evolution, if when we were kind of hunters and foragers, if we were going to eat like a plant or a berry or something or a fruit, that is basically our warning that we're about to eat something that is very poisonous to us. So if something tastes bitter, it's usually supposed to be a sign that, hey, you're not supposed to be eating what you're about to eat. Well, obviously, shows how smart we are, right? So now we eat all kinds of things that have bitter taste for it, and hey, we're happy about it, right? Interesting, right? So, and then the last taste, umami, which is, it's not a literal translation, I don't think, but it's a Japanese word that essentially means savory or meaty. So this is going to be activated by amino acids. So if you eat something that's high in proteins, particularly the amino acid glutamate, you're going to get kind of that savory, meaty flavor to it. So before we really delve into how these types of molecules activate the receptors, we should take a very, very brief look at the anatomy of the tongue, particularly the taste buds. So each taste bud, of which there are many spread throughout the tongue, each taste bud is a big kind of compact barrel of very columnar epithelial cells. Now, at the very tips of these epithelial cells, you'll recall the epithelial cells are polarized. So at the very tips, the apical side, are specialized cilia structures called taste hairs. It is at these cilia where the taste receptors are located. So if you kind of look up here, you've got the surface of the tongue up here, you've got these taste cells right here. on the surface of the tongue, that's going to be where the saliva is. So this kind of underlies the importance of getting your food molecules dissolved in the saliva, because that's going to be what allows those ligands, those food molecules, to interact with the receptors here on the taste cells. So from there, once you get that interaction to occur, different types of cells that are found within the taste bud are responsible for sending a message about a particular type of flavor. So you can see that this whole taste bud is connected via a sensory neuron axon fiber that should form a part of either the facial or glossopharyngeal nerve that carries that information to the thalamus and then eventually to the gustatory cortex. Now, here's the idea. So I mentioned the facial nerve and the glossopharyngeal nerve. So the idea here is that basically, each type of cell carries information about one particular flavor. It's often been thought that one taste bud carries information about one flavor. Well, that's not true. A single taste bud can actually carry information about many different flavors, all 5 of them if you want. But if that's going to be the case, to carry all 5 flavors, that taste bud has to have one of each of the 5 different types of taste cells. There are salty taste cells, there's sweet taste cells, and sour taste cells, and so on. Okay, so what we're interested in here is for each one of these ligand receptor combinations, whether it's sodium and the salty receptors, hydrogen ions and the sour receptors, sugars and the sweet receptors, and so on, for each one of these, we need to be aware of how each taste cell is going to get depolarized. Because, as always, this depolarization is going to be necessary in order to get an action potential carried through that sensory nerve fiber so that we can get that afferent taste information delivered to the brain. Now, in each case, the depolarization is going to work slightly differently. So let's start with the first 2, salty and sour. This is going to be kind of the easiest and simplest way to understand this. So basically what's happening here is that there's not so much an activation of a receptor so much as basically these guys are just going to move through open ion channels. So any salty stuff that contains sodium, well, that sodium is going to just go through an open ion channel and depolarize the cell that way, right? So you've got positive charges moving inside the cell. By definition, that depolarizes the cell, and then the taste cell will release neurotransmitter onto the sensory neuron, and we get an action potential that way. So nothing complicated there. It's just, hey, we've got all this sodium, let's let it into the cell, get our depolarization, and that's that. Same thing with sour taste. Hydrogen ions are also positively charged, so if they are let into the intracellular fluid, that will cause depolarization as well, and we release neurotransmitter the exact same way, and we get our action potential. So those are the 2 simplest. Now let's move on to sweet umami. Now, there's a note here. Sweet and umami receptors are absolutely 100% not the same. So the only reason in this little figure here, the only reason sweet and umami are kind of lumped together is because their signal transduction mechanism is almost exactly the same, but the receptors are different, meaning sugars do not activate umami receptors and amino acids do not activate sweet receptors. The only reason they are shown kind of in the same figure here is because their signal transduction mechanism is almost identical. Now, sugars and amino acids are not electrically charged. At least they're not supposed to be at neutral, at kind of physiological pH. So you can't just import them into the cell and expect to get depolarization. So we've got to get something else going on here. So what they are going to do is they are actually going to bind to G protein coupled receptors. That is going to activate an internal second messenger cascade, which will close some of those potassium leak channels that we've talked about before. And from the very act of no longer allowing potassium to escape the cell at a certain rate, that itself will cause depolarization, and we then get an action potential. And then finally, for bitter flavors, which are activated by alkaloid compounds, in this case we're looking at quinine, this is another G protein coupled receptor mechanism. We activate a second messenger cascade, and instead of actually depolarizing the cell, we actually don't depolarize the cell here. We are actually going to release calcium that is stored up in the endoplasmic reticulum, and this is going to itself trigger the release of neurotransmitter. So what you need to release neurotransmitter is essentially calcium. So basically, instead of getting our calcium from voltage-gated calcium channels like you see with the salty and sour, we just let calcium out of the ER and we get the neurotransmitter release that way. So let's take a little pause here to look at this figure one more time so that we can make sure we're appreciating what's going on here with taste. So we mentioned that afferent information regarding gustatory stimuli is carried by two different cranial nerves. We've got the facial nerve for kind of the anterior two-thirds of the tongue, and then the glossopharyngeal nerve for kind of the back third of the tongue. Both of these cranial nerves carry this afferent information into the brainstem. That information is deflected up to the thalamus, as afferent information tends to be, and then the thalamus will have cell bodies that project their axons to the gustatory cortex right here, and that's going to be the processing center for that taste information. And as was mentioned on a previous slide, I didn't really make a big deal of it, but this afferent information can also be shared with the limbic system, which, as we know, is associated with kind of fear and memory and emotion. So the things that we taste, the food that we eat, can actually evoke kind of emotional responses in us, which is very interesting. All right, so now let's move on to smell, or olfaction. And we're not going to spend too terribly much time talking about this because really it's very, very similar to what we saw with taste. Namely, smell is also mediated by chemoreceptors, except these chemoreceptors are not on the tongue. They are actually in the olfactory bulb, which is in the nasal cavity. So things are very similar here. These chemoreceptors are going to be activated by odorant molecules. In fact, they can even be activated by the very same molecules in the food that you eat. This is why food is often judged not only by how it tastes, but how it smells. And as you're going to see in your lab for this week, there's kind of a mixing of gustatory and olfactory information in the brain. So you kind of need both pieces of information processed in the brain together to really get a complete picture of how food really tastes, which is an interesting thing to talk about. But anyway, these chemoreceptors, similar to the taste receptors, they are located on the apical cilia of particular, basically, smell cells. We'll call these olfactory receptor cells. When these odorant molecules bind to these receptors, they will produce graded potentials or receptor potentials that should lead to action potentials carried through the olfactory nerve and into the brain, right? There is a similar requirement for the dissolving of these odorant molecules before they can bind to the receptors. In taste, the food molecules had to be dissolved in the saliva. Here, if you're going to smell something, the odorant molecules have to become dissolved in the mucus first. So we're not really going to make too much of a big deal about kind of the anatomy here. If you can grasp the information that's listed here in these three statements, you should be good, right? So not going to get too much into kind of the organization of kind of the primary and secondary sensory neurons here. We're not going to worry too much about that. But you can see the mucus and the pathway of air moving through the nasal cavity down here. So if the mucus is down here, then the tips or cilia of those olfactory cilia are going to be right here. So that's going to be where the interaction between ligand and receptor occurs, and then if everything goes well and the signal is strong enough, we should get action potentials carried up through here and then here through the olfactory nerve and into the brain. So if we look at this again, this time we only have one cranial nerve that carries olfactory information, in this case the olfactory nerve. It is cranial nerve number 1 because you can see it is closest to the anterior or front part of the head, carries this olfactory information, and as I'm sure you are aware by now, because we've mentioned it a lot, olfactory information does not project to the thalamus. It goes straight to the gustatory cortex, the olfactory cortex. All right, so that is going to do it for this particular video on taste and smell. Thank you for your attention, and next time we are going to start talking about audition, which is hearing, and then after that we'll finish up the chapter by talking about vision. Hope to see you next time. Bye-bye.